Hello dear students, in the last lecture we learned structure of a bacteria and today we will talk about the nutrition and reproduction of a bacteria. So let's get started. So we should know what kind of a nutrition bacteria needs. Bacteria need enough suitable nutrients for its growth and reproduction. And these nutrients are carbon source, nitrogen source, electron donors and trace elements. These are necessary for the growing bacteria. Then bacteria are extraordinarily diverse in their specific nutrient requirements. See, bacteria are very specific according to their nutrition as per their habitat. These bacteria are classified into various forms, so they are ordinarily diverse in their specific nutrient requirements. Means some kind of bacteria feeds on dead organic material, some kind of bacteria can feeds on living cells. So these bacteria are extraordinarily diverse. So let's see the nutritional types of bacteria. The various nutritional types of bacteria are discussed below. On the basis of nutrition, bacteria are divided basically into two groups: autotrophic bacteria and heterotrophic bacteria. Autotrophic bacteria means such bacteria can synthesize their own food, and heterotrophic bacteria are which cannot produce their own food but can feed on other organisms. So let's see autotrophic bacteria. These are some bacteria which are able to synthesize organic compounds necessary for their structure and metabolism from simple inorganic compounds. It is known as autotrophic bacteria. These autotrophic bacteria are thus of two types. So it may be photosynthetic and it may be chemosynthetic bacteria. The former are also called photosynthetic autotrophs and latter non-photosynthetic autotrophs. So let's see the first type, photosynthetic bacteria. They can prepare their own food by photosynthesis, and these are green sulfur bacteria, purple bacteria and non-sulfur purple bacteria. So what are green sulfur bacteria? These are the anaerobes, means they perform photosynthesis without evolving the oxygen, and they contain chlorobium chlorophyll pigments. These green sulfur bacteria use H2S or other sulfide as the electron donor. So these are the green sulfur bacteria. Second one is the purple sulfur bacteria, and they have photosynthetic pigment bacteriochlorophyll, and the electron donor in purple sulfur bacteria is also a H2S. Then third one is the non-sulfur purple bacteria. They do not use sulfur but make use of organic acids as substrates, and its photosynthetic pigment is bacteriochlorophyll. Let's see chemosynthetic bacteria, second type. These are non-photosynthetic autotrophs, means these are the nitrifying bacteria which lack pigments. They get energy for food synthesis by the oxidation of certain inorganic substances such as ammonia, nitrites, ferrous iron, etc. See, photosynthetic bacteria prepares their own food with the help of photosynthesis, but chemosynthetic bacteria, there is an involvement of different kinds of oxidation of chemicals takes place, and these chemicals, it may be ammonia, it may be nitrate, it may be nitrite, hydrogen sulfide and other inorganic compounds. So chemosynthetic bacteria, these are the non-photosynthetic autotrophs. Photosynthetic bacteria, there is an involvement of different kinds of pigments, but in chemosynthetic bacteria there is no pigments are present, and there are no pigments, and that's why they feed on the oxidation of certain inorganic substances. So this was about the chemosynthetic bacteria. These bacteria are having certain names, means Some are the sulfur bacteria, some are the hydrogen bacteria, nitrifying bacteria. So these names have been given to them on the basis of kinds of oxidation, means kinds of substances of which oxidation takes place. So see the first one, sulfur bacteria. These bacteria obtain energy by oxidation of elemental sulfur, or it is also known as H2S, and its example is Thiobacillus and Bagiata. Second one is the iron bacteria. These bacteria inhabit water and obtain energy by oxidation of ferrous compounds into ferric forms, and the example of iron bacteria are Thiobacillus ferrioxidans. Third one is the hydrogen bacteria. These convert hydrogen into water, and the example of hydrogen bacteria is Bacillus pentotrophus and Hydrogenomonas. Last one is the nitrifying bacteria. These bacteria get their energy by oxidation of ammonia and nitrogen compounds into nitrates. Means such nitrifying bacteria are very beneficial for the trees, means they does the nitrogen fixation. So this was about the autotrophic bacteria. Now we will discuss the heterotrophic bacteria. Heterotrophic bacteria are, they derive their energy from organic compounds. They may be aerobic or anaerobic, and these are parasitic, these are saprophytic, and they may be symbiotic. So what are parasitic? They derive their food from living organisms. Saprophytic bacteria are, they feed on dead and decaying organic material, and symbiotic, they live in symbiotic association with other organisms, means there is no harm to the other organism. So let's see one by one. Number one is parasitic bacteria. These bacteria obtain their nutrition from the tissues of the host on which they grow. These are the bacteria which are pathogenic. They live on the tissues of the host, and these parasitic bacteria which causes various diseases in plants and animals are known as pathogens, and the example of these parasitic bacteria are Bacillus thiophorus, Bacillus anthracis, Xanthomonas, etc. So these are the parasitic bacteria which lives on the other living host organisms and feed on them for their nutrition. The second type of these heterotrophic bacteria is saprophytic bacteria. Saprophytic bacteria obtain their food from the dead and organic decaying matter, such as leaves, fruits, vegetables, meat, animal feces, leather, humus, etc. These kind of bacteria perform fermentation and putrefaction process where carbohydrates and proteins are broken down into simple compound. Means lactic acid fermentation is common process where milk is converted into curd by the lactobacillus bacteria. So these are the saprophytic bacteria. Now let's see the third type of this heterotrophic bacteria, and that is symbiotic bacteria. Symbiotic bacteria live in close association with other organisms as symbionts. They are beneficial to the organisms, means there is no harm to the other organisms. The common examples are the nitrogen fixing bacteria, and these are Bacillus radicicola, Bacillus azotobacter, Rhizobium, Clostridium, Rhizobium. All these are the symbiotic bacteria, nitrogen fixing bacteria. See, for the survival, plant needs nitrogen, but plant directly cannot absorb this nitrogen. So these nitrogen fixing bacteria, these bacteria fixes the nitrogen, means there is a conversion of nitrogen into nitrates or nitrites, and then this plant will absorb this nitrogen in it for its nutritional value. Means indirectly they are giving nutrition to the plant. So this is the symbiotic bacteria. Now let's see these terms. different kinds of terms. Number one is the phototrophs, means these derive energy from sunlight. Then chemotrophs, they obtain energy from chemical reactions. Then autotrophs are they synthesize all organic compounds. Heterotrophs means unable to synthesize and depend on performed organic compounds. So these are the different terms which you should learn and you should remember all the time. Now this was all about the nutrition. Now we will talk about the reproduction of a bacteria. So the first one is the sexual reproduction of a bacteria. Actually in bacteria, this is the genetic recombination. True sexual reproduction is absent. So there is a transformation of genetic material from one bacteria to another takes place. So that is why it is known as the sexual reproduction, but it is actually the genetic recombination, and these are of three kinds. It may be conjugation, it may be transformation, and it may be transduction. So let's see one by one. Number one is conjugation. One bacterium connects itself to other through a protein tube structure called a pilus. If you remember the structure of bacteria, you will understand this pilus. Genes are transferred from one bacterium to the other through this tube, and such tube is known as the conjugation tube. See this diagram, in which you can see this is the pilus. There is a formation of a tube in between two bacteria, and in the third step you will see that there is some kind of a fragmentation takes place of the plasmid, and this fragment will cross this pilus. Actually now it is not a pilus, it is the conjugation tube, and then this pilus will form this fragment will produce this plasmid. Means all the genetic information is present in this plasmid. So the information is crossing, it is transforming from one bacterium to the another bacterium. So this is the first type of the sexual reproduction of a bacteria, and that is conjugation. Now we will see the second type, and it is the transformation. What is transformation? The bacterium binds the DNA and transport it across the bacterial cell membrane, and the new DNA is then incorporated into the bacterial cell's DNA. In this kind of reproduction, the molecules of a DNA move from the donor to the recipient cell through the liquid medium. Means genetic information is get transformed. So that is why it is the transformation. I will explain with the help of this diagram. See, if you can, this is the DNA fragments from donor cells, and these fragments are they taken up by the another bacteria, and this process takes place through the liquid medium. If you see the chromosomal DNA of this recipient cell, these are small letters A, B, C, D, E. Okay, now these fragments are having the capital letters A and B. So one of the fragment is get incorporated into this recipient cell. In the next step, what will happen? There will be formation of recombination. Recombination will occur between the donor DNA and recipient DNA, and see, this is the genetically transformed cell. Means these are the fragments from another donor cells, but it get incorporated into the chromosomal DNA of the recipient cell. If you see small a, capital B, then small c, d, e. So this is the transformation. There is a transformation has been takes place. This was about the transformation, and the last type of this sexual reproduction of bacteria is the transduction. Let's see what is transduction. In transduction type of reproduction, there is an involvement of exchanging of bacterial DNA through bacteriophages. What are bacteriophages? Bacteriophages are the viruses which infects the bacteria. We have seen when we have discussed about the viruses. So there is involvement of this bacteriophages in transduction. Here you can see this is the bacteriophage. It will transfer its own genetic material into the bacteria. Means see, we have discussed the structure of a viruses, which is made up of protein and genetic material. It may be RNA, it may be DNA. So what it will do, this is the virus, it is bacteriophages, but it is the virus. And why it is bacteriophage? Because it is infecting the bacteria. Now the protein coat will be extracted outside, but it will transfer its own DNA into the bacterial DNA. These bacterial DNA and virus DNA, it will get fragmented, and then it will produce its own copies inside the bacteria. So this was about the transduction, which takes place with the help of the bacteriophages. Now let's see the asexual reproduction. Bacteria can reproduce by binary fission, budding, then conidia formation and endospore formation. So let's see one by one. Binary fission. In binary fission, what happens? This is the cell wall and this is the plasma membrane of a bacterial cell. Number one, cell elongate DNA is replicated. As the cell get elongated, the DNA is get replicated. Then cell wall and plasma membrane begin to constrict. Means there is a formation of the invagination. In the third step, cross wall falls, completely separating the two DNA copies. Means these two DNA copies are get separated, and then in fourth, finally the cells are separated. So this is how the binary fission takes place. Means the elongation of cell takes place, because of which DNA will be replicated. There will be formation of another DNA from the previous one. Then there will be the formation of invagination, means from which these two cells this one cell will get separated from the portion of the invagination, and then there will be formation of two cells of the bacteria. Now the second type of this asexual reproduction is budding. Budding of bacteria, cells genetic information is duplicated, creating an identical copy or clone of the original cell. As the new cell pinches off or buds from the surface, a bud scar is produced. From the diagram you can see how the bud formation is taking place and how these bacterial cells are getting separated from each other by just formation of the bud. So this is the budding of bacteria. Then asexual reproduction can take place by the formation of conidia. It occurs in members of certain families belonging to Actinomycetes. Example, Streptomyces. The ends of the filaments become separated. In the diagram you can see separated from a chain of conidia similar to those produced by true fungi. These conidia can resist desiccation, means similar to endospore, but they cannot resist high temperature. So this is the conidia formation. Means many filamentous bacteria forms chain of small spherical spore like conidia at the tip of these kind of filaments, and they develop by the formation of transverse wall at the tip of the filaments. Next one is the endospores. These endospores are intracellular resting cells. These endospores are formed in some gram-positive bacteria. Here you can see this is the endospore and this is the parent cell of this endospore. Now there is the formation of this exporium, core wall, spore coat, plasma membrane, and this is the cortex. And from these endospore, new bacterium will be produced. This endospore, it is the oval or spherical and may be larger or smaller than vegetative cell. It is made up of central portion, the core, and covered by a core membrane. The multi-layered wall around core is the cortex. Outer to the cortex is a spore coat made up of inner coat and the outer coat. So this was about the asexual reproduction of a bacteria. In the next lecture we will see the role of a microbes, and this that will be the last part of our unit. So see you in next lecture. Thank you so much.